Dear students, I welcome you to the e-learning program through ePG Pathshala. I am Dr. K. Pandima Devi, working as Associate Professor in Department of Biotechnology, Alagappa University, Karaikudi. In this session, I am going to explain to you about the role of antioxidants in health and diseases. We will discuss about the role played by the antioxidants in protecting the human system from the diseases caused by oxidative stress. Free radicals are produced in the body through both endogenous and exogenous sources, and they play a major role in mediating many diseases like cardiovascular disease, neurodegenerative disorders, cancer, and so on. This session will help you to understand the role of antioxidants in fighting the oxidative stress mediated disorders which are induced by the free radicals. After completing this course, you will be able to understand the damage caused by free radicals to the human system, appreciate the neutralizing effect of antioxidants against free radicals, and know the role of antioxidants in maintaining health. Before discussing on the role of antioxidants in health and diseases, we will first see about how free radicals are generated in our system. Free radicals are atoms or molecules which have an unpaired electron in its outermost shell, due to which they are unstable and highly reactive. The free radicals may be generated in the system either through endogenous or exogenous sources. Endogenous free radicals are the ones which are generated inside the cells during the normal metabolic process of the cell, including autooxidation, electron transport system, enzymatic production by peroxidases, cyclooxygenases, NADH oxidase, transition metal mediated, and so on. Free radicals are generated not only inside the cytosol, even the cellular organelles like mitochondria, lysosomes, and peroxisomes contribute for the production of free radicals. Free radicals production becomes deliberate during certain processes like elimination of the foreign pathogens by phagocytic cells like macrophages, monocytes, neutrophils, and eosinophils. If you take the exogenous free radicals, they are the ones which are produced during exposure to UV rays, toxic heavy metals, environmental toxins like industrial waste, and cigarette smoke. Now, if you see the types of free radicals, the major free radicals and the reactive molecules which are produced in our system includes reactive oxygen species, otherwise termed as ROS, and reactive nitrogen species, otherwise termed as RNS, which includes superoxide anion, hydroxyl radical, singlet oxygen, peroxyl radical, alkoxyl radical, hypochlorite, hydrogen peroxide, nitric oxide, and peroxynitrite. If the free radicals are not eliminated effectively, they target the macromolecules of the cell, which lead to the lysis of the cells. Peroxidation of the membrane lipids by the free radicals affects the fluidity of the membrane. Similarly, free radicals damage the proteins and lead to denaturation of proteins and induce mutation by modifying the DNA bases. If the generation of free radicals is not regulated, it can induce a variety of chronic diseases including cancer, neurodegenerative disease, as well as aging process. So you would have now understood that the release of free radicals induces a damaging effect in the cells. To protect against the harmful effects of the free radicals, our system is equipped with an array of defensive molecules called as antioxidants, which either limit the formation of free radicals or break the chain breaking free radical reactions. The antioxidant defense system includes enzymes like catalase, superoxide dismutase, vitamins like vitamin C, E, and even biomolecules like bilirubin, uric acid, and so on. These antioxidants, which may be either endogenous or exogenous, which is taken through the diet, plays a major role in neutralizing the effect of oxidative stress and in further preventing or treating various diseases. In certain cases, for the treatment of certain chronic diseases like cardiovascular disease, diabetes, and cancer, it becomes essential to change the lifestyle pattern by supplementation with exogenous antioxidants through diet, which can complement the action of endogenous antioxidants. Let us now see in detail on the role of antioxidants in the treatment of major diseases like lung disorders, cancer, aging and age-related disorders, cardiovascular disease, and liver disorders. First, we will see about lung disorder. How is oxidative stress generated in lung diseases? Oxidative stress is thought to play a role in a variety of lung diseases including asthma, lung cancer, chronic obstructive pulmonary disease, acute lung injury, and so on. Production of reactive oxygen species seriously alters the cell membranes and other structures such as proteins, lipids, lipoproteins, and DNA. Lung is constantly exposed to both endogenous oxidants through mitochondrial electron transport chain or phagocytosis process and exogenous oxidants through exposure to toxins, cigarette smoke, air pollutants, and drugs. The major phagocytic cells present in large numbers in the lungs are the alveolar macrophages, which comes as the first line of defense against lung infections. The NADPH oxidase enzyme present in the membrane is the major source of the reactive oxygen species. Eosinophils, which contain the NADPH oxidase, also contributes to the formation of reactive oxygen species, especially during certain allergic and inflammatory conditions. During pulmonary disease, the peripheral monocytes, which are attracted by the inflammatory cytokines, differentiates into macrophages and produces superoxide anion by the enzyme xanthin oxidase. During lung injury, an increased activity of xanthin oxidase has been detected in the bronchoalveolar lavage fluid. In chronic obstructive pulmonary disease, which is mainly induced by smoking, iron plays a role in development of oxidative stress. The alveolar macrophages and the lung lining fluids obtained from the cigarette smokers contain more iron concentration than the non-smokers. Normally, iron is bound to various iron-binding proteins like transferrin, ceruloplasmin, and ferritin, whereas in chronic obstructive pulmonary disease patients, iron is released from these proteins which contribute to the production of reactive oxygen species. Now, how does the antioxidants protect against the lung diseases? The antioxidant defense system in the pulmonary system includes both enzymatic and non-enzymatic systems. The major antioxidant system includes the enzymes superoxide dismutase, catalase, glutathione peroxidase, thioredoxin, and glutaredoxin. Three forms of superoxide dismutase are involved, like manganese superoxide dismutase, which is located in mitochondria, copper zinc superoxide dismutase residing in the cytoplasm, and extracellular superoxide dismutase, which lines the blood vessels. The enzyme catalase found in alveolar macrophages, pneumocytes, and lung fibroblasts exerts its antioxidant function by reducing the hydrogen peroxide into water. Glutathione peroxidases mainly defend the alveolar epithelial cells against oxidative stress such as hyperoxia-induced liver damage. Thioredoxin, which is a small redox protein mainly present in the bronchial epithelium and the alveolar macrophages, protects the tissues against exogenous antioxidants. The vitamins like vitamin C, E, and also beta carotene present in many fruits and vegetables can also protect the lung from smoking or pollution mediated oxidative damage. In smoking induced chronic obstructive pulmonary disease, decreased activity of the nuclear erythroid 2 P45 related factor 2, which is called as NRF2, a redox sensitive transcription factor, has been noticed. Since activation of NRF2 expression could trigger the expression of antioxidative and cytoprotective enzymes, antioxidants with NRF2 activation can be used as an approach for treatment of chronic obstructive pulmonary disease. Next, we will see the role of antioxidants as therapeutic agents for treatment of cancer. Oxidative stress has been linked with the initiation and progression of various types of cancer. The free radical induced DNA oxidation includes modification of the bases, DNA crosslinks, and break in the single or double-stranded DNA. This leads to errors in replication, instability of the genome, and induction of the signal transduction pathways. The most common damage induced by free radicals formed through chemical carcinogens are the formation of the hydroxylated bases by the hydroxyl radicals. These adducts which are formed induces mutation in the genes alters the transcription of the genes, and interferes in the normal growth of the cells. An example is the formation of 8-hydroxyguanine, which is both a carcinogenic and a mutagenic agent. In certain types of cancer, like breast cancer, even 8 to 17-fold increase in 8-hydroxyguanine has been reported when compared to non-malignant tumor cells. The changes in DNA induced in cancer patients may be genetic or due to exposure to certain environmental factors like tobacco, pollution, drugs, radiation exposure, metal poisoning, and so on. These substances, which lead to the development of cancer, are called as carcinogens. One of the mechanisms through which carcinogens mediate the initiation of cancer is through production of oxidative stress. For example, increased levels of metals like iron, copper, chromium, lead, and so on induce toxicity and carcinogenicity mediated through the generation of reactive oxygen species and reactive nitrogen species. The carcinogens induce modifications in DNA bases and also enhance the peroxidation of the lipids. Metals like chromium, once they enter inside the cells, in the presence of reductants present in the cell like glutathione, gets converted to other forms which can form adducts with DNA and also induce oxidative damage. Even oxidative stress associated with chronic inflammatory conditions is associated with the increased incidence in the development of certain types of cancers. During the inflammatory conditions, the mast cells and leukocytes, which are recruited to the site of damage, releases increased levels of reactive oxygen species. For example, the hydrogen peroxide, which is produced during the dismutation of superoxide anions, play an important role in the carcinogenesis process since they can cross the cell membranes and induce many types of cell injury. Since free radicals has a major role in creating the tumor microenvironment, as cancer progresses, the levels of antioxidants in the cells decrease to a greater extent. Many types of carcinogens, UV light, ionizing radiation, have been observed to inhibit the activities of antioxidant enzymes like catalase and superoxide dismutase. Hence, supplementation with antioxidants may offer a protective mechanism for the treatment of cancer. For example, vitamin E or alpha tocopherol, which is a lipid-soluble antioxidant, protects the lipids against the peroxidative damage. Alpha tocopherol succinate, the analog of alpha tocopherol, is more effective in inducing apoptosis in cancer cells. Also, high consumption of the antioxidant yellow pigment curcumin, which is isolated from turmeric, is linked with the low rate of colon cancer. It induces apoptosis in the cancer cells by suppressing the activity of the anti-apoptotic protein BCL2. Natural green tea, which is consumed in large amount in Asian countries, contains a variety of antioxidants including epigallocatechin 3 gallate. This antioxidant compound induces apoptosis in cancer cells by activating the caspase enzyme. Not only natural antioxidants, even synthetic chemical antioxidants like butylated hydroxytoluene and butylated hydroxyanisole have also been found to inhibit the development of carcinogen-induced tumors. Next, we will discuss about the role of antioxidants as therapeutic agents for treatment of aging and age-related disorders. Aging is a multidimensional, irreversible accumulation of physical, environmental, and social changes. It is one of the major risk factors for the development of many diseases including cardiovascular diseases, stroke, cancer, and arthritis, and several neurodegenerative diseases. Oxidative stress and mitochondrial dysfunction are the two major factors which contribute to the aging process. The free radical theory of aging hypothesizes that oxygen-derived free radicals are responsible for the age-related damage at the cellular and tissue levels. According to the theory, oxidative stress attacks mitochondria and leads to increased oxidative damage. The mitochondria lose their functional integrity and release more oxygen molecules, which culminate in the accumulation of dysfunctional mitochondria with age. Since mitochondria plays a crucial role in regulation of apoptosis, dysfunction of mitochondria during the aging process may be the reason for increased cell death caused by apoptosis. The increased levels of 8-hydroxy-2-deoxyguanosine, a biomarker of oxidative DNA damage, has been observed in mitochondrial DNA in the aged brain. The mitochondrial theory of aging system reveals that in age-related diseases, there is an increase in the level of both oxidative damage and mutational load of mitochondrial DNA. One of the major free radicals is superoxide anions, which is produced by aging mitochondria due to increased mitochondrial DNA damage in the aging process. Chronic inflammation, which may be mediated through oxidative stress induced by environmental or lifestyle factors, has also been linked with aging. Oxidative stress has been associated with all types of age-related neurodegenerative disorders like Parkinson's, Alzheimer's, and Down syndrome. In these diseases, neurons of the human brain are affected and there is a loss in the nerve cells from brain and the spinal cord. The glial cells which are present in the brain generates ATP for the normal functioning of the brain, for which it consumes large volumes of oxygen and glucose. This makes the brain cells more susceptible for oxygen overload, which leads to the increased generation of free radicals. We will now discuss on the role of antioxidants in combating aging and age-related disorders. Since aging imposes oxidative stress in the body, the antioxidant enzymes are significantly altered during aging. Dietary supplementation with antioxidants like vitamins decreases oxidative stress and prevent age-related changes in the central nervous system. For example, supplementation with vitamin E has been considered as an effective treatment for Alzheimer's disease since it improves the cognitive function. Curcumin, which is a more potent antioxidant, scavenges free radicals and reduces oxidative damage in Alzheimer's disease. Telomere, which is the end cap of segment of our DNA, gets shortened during cellular aging and other neurodegenerative disorders. Curcumin slows the aging process by increasing the expression of telomerase and thus preserving the length of telomere. Next, we will discuss about how antioxidants serve as therapeutic agents for treatment of cardiovascular disease. Cardiovascular diseases, which mainly affect the heart and blood vessels, include stroke, hypertension, atherosclerosis, and acute myocardial infarction, which we commonly call as heart attack. Apart from the other risk factors like increase in cholesterol level, blood pressure, smoking, and diet, which leads to the development of cardiovascular disease, free radicals either directly or indirectly contribute to the oxidative damage in heart tissues. Either the intracellular reactive oxygen species, which are formed from mitochondria and xanthine oxidase, or the extracellular reactive oxygen species formed from the neutrophils and macrophages, serve as sources of reactive oxygen species which are involved in the myocardial cell damage. Antioxidants play a major role in treatment of many cardiovascular diseases. For instance, in the cardiovascular system, the overproduction of free radicals leads to endothelial dysfunction, which may be mediated through the peroxidation of lipids, loss of nitric oxide bioactivity, or the other factors like inflammatory responses. The antioxidants break these chain reactions by donating electrons to the free radical molecules. Many dietary plants offer a protective function in the treatment of cardiovascular diseases. Hence, adequate intake of nutrition which supplies appropriate amount of antioxidants play an important role in the prevention of cardiovascular disease and can also help in improving the symptoms of the disease. Intake of vitamins like vitamin C and E improves endothelial function in patients with hypertension. Supplementation of vitamin D is beneficial to endothelial function, blood pressure, and cardiac hypertrophy in humans. Polyphenols, which are the most common constituents of plants present in the human diet, contributes to the prevention of many diseases including cardiovascular diseases. One example is administration of pomegranate juice to atherosclerotic mice reduced lipid peroxidation and also lowered the accumulation of cholesterol and the further development of atherosclerosis. Next, we will see about how antioxidants help in protecting against liver diseases. Reactive oxygen species has been observed to play a vital role in the development of many types of liver diseases which causes damage to the liver like cirrhosis, hepatitis, liver cancer, alcoholic liver disease and so on. Various endogenous and exogenous factors are believed to play a role in releasing the free radicals in the liver. For the non-phagocytic cells like hepatocytes, the major source of free radicals is the mitochondria, which is produced during oxidative phosphorylation process. The enzyme NADPH oxidase generates superoxide anion radicals as a defense against invading pathogens. However, when this process is interfered, it leads to the overproduction of superoxide anions which causes damage to the cell. The superoxide anions which are produced later generates the other free radicals like hydroxyl radicals, hydrogen peroxide and the like. The endoplasmic reticulum also contributes to the generation of free radicals in liver diseases through the cytochrome P450 enzyme. Free radicals are generated in almost all the chronic diseases in the liver, which activates many oxidant dependent signaling pathways and leads to the apoptosis of the liver cells. Apart from mitochondria and endoplasmic reticulum, the neutrophils and Kupffer cells which infiltrates into the liver during any inflammatory process or injury also contributes for the release of reactive oxygen species. Since mitochondria acts as a major source of reactive oxygen species in liver diseases, antioxidants which are specifically taken up by the mitochondria is considered for treatment of liver diseases. Example of such a compound is mitoquinone, which has a ubiquinone component for mediating the antioxidant activity and a lipophilic cation to facilitate its accumulation inside the mitochondria. Antioxidant therapy for treatment of liver diseases include intake of vitamins which possess antioxidant activity like vitamin A, E and C. During liver diseases, the level of S-adenosyl methionine, which acts as a precursor of the antioxidant molecule glutathione, generally decreases. Hence, S-adenosyl methionine is administered for the treatment of chronic liver diseases. Also, drugs obtained from alternative sources like silymarin, which is extracted from the milk thistle or Silybum marianum, are used from ancient time for the treatment of liver diseases. It contains a mixture of polyphenols and flavonoids, with the main component as silibinin along with other flavonoids such as silicristin, silidinin, silibinin and isosilibinin. Silymarin has been proven to possess antioxidant activity and has a beneficial role in protecting against oxidative stress induced liver injury, liver cirrhosis and chronic hepatitis induced by virus. In this session, we have discussed about the role of antioxidants in health and diseases. You would have now understood that excessive free radicals are produced in many diseases from both endogenous and exogenous sources. Since the free radicals causes excessive damages to the macromolecules like DNA, proteins and lipids, many endogenous and diet-derived exogenous antioxidants eliminate the free radicals and prevents the free radicals mediated damage. We will be discussing about the classification and types of antioxidants in the subsequent module.